There has always been a huge effort to develop less toxic regimens, less toxic than an auto transplant, and also look forward to doing something better in patients who are transplant ineligible. So with that in mind, now nearly 4 years ago, there were three pivotal studies that were designed, namely Zuma-1, exploring axicabtagene, Juliet exploring tisagenlecleucel, and Transcend exploring lisacabtagene in a heavily pretreated relapsed refractory large B-cell lymphoma population, and based on the excellent outcomes that were demonstrated, including in patients who had failed 2 lines of treatment or more, including patients who had failed an auto transplant, the FDA approved these CD19 CAR T-cell therapy constructs in patients with relapsed refractory large B-cell lymphoma. Encouragingly, we now do have long-term follow-up data from Zuma-1, Transcend, as well as Juliet that actually support a curative potential of CD19 CAR T-cell therapy in relapsed refractory large B-cell lymphoma after failure of 2 lines of treatment. Across the 3 studies, we now have a 5-year survival that is in the range of 30 to 43%. The newest kid on the block has been the emergence and the FDA approval of anti-CD20, CD3 bispecific T-cell engagers. 2 bispec antibodies have been approved by the FDA for patients who have failure of 2 lines of treatment with large B-cell lymphoma. Glofitamab and epcoritamab are now FDA approved. There are key differences between the structure as well as the administration. So glofitamab is a 2:1 binding. Epcoritamab is a duo-body construct. Glofitamab is given as an IV infusion. But I think the merit of glofitamab is in the fact that it is fixed duration treatment. It's only given for 12 cycles. Epcoritamab, on the other hand, is easy with regards to administration because it's sub-acute administration, but it's given indefinitely or until treatment progression. The overall response rate was between 50 and 60%. The complete response rate across the 2 constructs was in the range of 39%, and the complete response rates were not affected by prior CAR T administration, and it was about 35% across the 2 studies. So I think an important question to answer is in the contemporary era as of current practice would be what to choose if patients have relapsed after 2 or more lines of treatment, because we have the ability to give them CD19 CARs, we have the ability to give them bispecs. And I do believe that, one can make a case for each. But if we were to talk about just the case for CAR T-cell therapy prior to bispecific T-cell engagers in the 3rd-line setting, I think I favor that because there's a potential for cure in about 30 to 40% of patients as a function of long-term follow-up at 5 years now. It's a single infusion, 1 and done. Higher response rates, higher progression-free survival, especially in the refractory large B-cell lymphoma population. Cytotoxicity is completely independent of innate immunity, and response is independent of antigen expression. I think the case for bispecifics is primarily built around the fact that it is off the shelf. As a result, immediate treatment initiation is possible. There's no need to bridging treatment. There's no need to commitment to lymphodepletion chemotherapy. It has the potential to be administered in the community setting, but a lot of education is required because with bispecific antibodies, you can still get cytokine release, and the community practitioners have to be able to manage cytokine release syndrome. Generally lower rates of cytokine release and very rare neurotoxicity. So overall, I think in conclusion, I do believe that both of these constructs are highly efficacious, but in the 3rd-line setting, I prefer CARs followed by bispecifics. However, if patients have received CAR T-cell therapy in the 2nd-line setting, then obviously my preference is bispecs. I think this debate is not going to last for too long because both of these constructs are being explored in earlier lines of treatment. So I think at this point in time, though, shared decision-making would be really important to determine what the patient and his or her family, the disease state, etc., would merit in terms of choosing between CARs versus bispecifics.